Hey gang, how are we doing today? Doing good, I see. Well, today we're going to start Chapter 1, Introduction to Bee Lifestyle. Reminder, we have our worksheet due tonight at 6 p.m., so try to remember to turn that in. I do have office hours afterwards in case anybody needs some extra help, but just try to remember that. Let's go ahead and get started. Our first topic for today is going to be, why do we make honey? As you know, we need food during the winter, and flying and to keep us going takes a lot of energy. So we have glucose stored in polymers called glycogen, and these polymers are easily broken down into glycolytic fuel. So if you look at the next slide, you see that we have our glycogen polymer. And remember, enzymes have very specific jobs as to what they do, and their names are really descriptive. In this case, our glycogen phosphorylase will attach itself to a glucose via reduction, and it will reduce the glycogen polymer as well, and that's how we get our individual monomers of our glycogen polymer. For our next topic, we're going to discuss, where do we get honey? Does anyone want to elaborate on this for me?
I do.
Go ahead, Laura.
Alright, so we get honey from flowers. So when we're flying through the air looking for food, strong scents can attract us to a certain type of flower. So these smells come from a variety of low molecular weight chemicals that are called plant volatils. And they're called this because they're so light that they easily evaporate and can get carried through the air. So there's many different types of these compounds. One of them is oxygenated VOCs, and that VOCs is volatile organic compounds, such as methanol, acetone, or methyl ethyl ketone. There's some various sulfur compounds found in different fruits. There's some furanocoumarins, which contain furan rings, which are 5 numbered rings with an oxygen within the ring. And then there's terpenes. So terpenes are a blanket name for volatile organic compounds that are responsible for characteristic scents that attract a lot of different pollinators, like bees, birds, and also butterflies. So the building blocks of all of these terpenes, it's a 5-carbon structure that's called isoprene. And so from that original structure, it can add on a lot of different chemicals and other functional groups to form different cyclic or acyclic compounds. And some of the most common ones are limonene, which is in citrus fruits. You have alpha and beta pinene, which is found in pine needles and pine trees. And there's linalool, which is found in lavender, which is a flower that attracts bees.
Okay, so for our next topic, we're going to go over, how do we make honey? Does anybody else know anything about this?
I do.
Thank you, Catherine.
So honey making is the backbone of bee society. Our brave forager bees go out into the wild and they look for flowers, and they gather the nectar from it, and it goes into their honey stomach. Nectar is 70 to 80% water, and it has sucrose in it. What the forager bees do is they bring all of that nectar back to the hive, and they give it to the processor bees. That nectar is passed from bee to bee, and after every single bee, more invertase is added, and this can catalyze a hydrolysis reaction. That can turn the long sucrose molecule into glucose and fructose, which are easier to digest. Once we eventually make that nectar into honey, the flavors can change depending on what the nectar source was, what type of flower it came from. There's a lot of water in the nectar, like I said, so that has to be evaporated eventually so that the honey doesn't go bad over time. So the way we get the water out is by spreading it across the honeycomb so that its surface area is maximized. And we fan our rings over it so that the water leaves. This is the reaction of sucrose being broken down. It's pretty simple, and it happens naturally when there's water present, but the invertase catalyzes it and makes it go a lot faster. Basically, that oxygen that's shared between glucose and fructose, that's where the bond is going to split. And the oxygen is going to go with the sucrose and take the hydrogen from the water, and the OH from the water is going to go onto the fructose molecule, and now you have 2 sugar molecules.
Thank you, Catherine. Okay, for our next topic, we are going to discuss, how does honey not spoil? Does anyone want to go over this for me?
I can.
Well, thank you, Laura.
Okay, so honey is pretty acidic. It's got three main acids in it, which are formic acid, citric acid, and then the major one, which is gluconic acid. And as the name suggests, gluconic acid is made from glucose. So the acidity from these three acids gives honey a pKa of around 4, which is pretty low. And that, along with the low water content from all the water getting evaporated out, makes it really hard for bacteria to survive in honey, so it won't spoil even after thousands of years. So the world's oldest honey is actually 5,500 years old. So producing gluconic acid, it starts from glucose. And there's an enzyme that catalyzes it. It's called glucose oxidase. And as you said before, they have really descriptive names. Glucose oxidase oxidizes glucose into gluconic acid. So the way the reaction works is that the enzyme will attack the OH group that's on the right side of the molecule, and it'll oxidize it into a ketone. And once it's in that ketone form, it'll undergo oxidative cleavage to form a carboxylic acid group on one end of the molecule and an alcohol group on the other. Hydrogen peroxide is also produced in these reactions.
For our next topic, we're going to discuss how can other species use honey. Does anyone want to go over the first little bit of this for me?
I can.
Amy, go ahead.
All right, well, the main reason that other species steal our honey is to eat it, and let's face it, honey is freaking delicious. But also honey is a humectant, which means it absorbs water from just its surroundings in general. And this is mostly because honey is hygroscopic. It's composed of a lot of sugars, and those sugars don't really contain a lot of water. And so if you leave honey just out unsealed, they can absorb water by osmosis, mostly into form crystals. Some species use this property of honey to make moisturizers, like soaps, lotions, things like that. But you have to be really careful when you do that because all of the sugars in honey can scorch at temperatures required for the normal saponification process. And that's why people often use aloe or other humectants that don't have as high a sugar content.
Yeah, that's really good. Honey is also actually a really good antioxidant. Species that consume honey use it for its antioxidant effects because honey contains a lot of polyphenols which engage in free radical mechanisms. This lowers the amount of free radicals in the body, which can be damaging to one's health. So if you look here, here's some common examples of some polyphenol antioxidants. You can see there's a lot of them within honey, which is why it is a good antioxidant. So in this mechanism, a free radical will engage with the pair-substituted alcohol, and what will happen is it reduces it, or it oxidizes it, so that the free electron will then delocalize itself around the ring. And we know positive charge delocalization makes a molecule very stable, which is why it's way better to be on these polyphenol rings than it is to be loose within the body. So the last thing that we're going to go over today is some of the dangers that are associated with collecting honey. Does anyone want to elaborate on this for me? Well, thank you, Amy.
I think the biggest danger associated with collecting honey nowadays is a pesticide known as a neonicotinoid. It's a water-soluble poison that has become really common since humans developed it in the 1980s and first started using it in the 1990s. And it's a water-soluble poison that humans will put in the soil and on their plants. So the plants will absorb it up as they absorb water through their roots, and then the poison becomes part of the plant's tissues. And then when the flowers become infected with the poison, bees will ingest it as they go to harvest the nectar. And a dose of 4 to 5 nanograms of some types of neonicotinoids is enough to kill a lot of honeybees. And even if in sublethal doses, the poison can kind of hinder a honeybee's ability to navigate and find routes to the flowers and food sources that you found before. And it can just overall reduce the resiliency of the colony in the winter. So over on the left, those are 3 common neonicotinoid pesticides. And over on the right is the structure of an acetylcholine molecule. acetylcholine is a neurotransmitter in a lot of just animals in general. And a lot of the functional groups on those neonicotinoid pesticides, as you can see, are very similar in structure to the amine, the active group on acetylcholine. So once a neonicotinoid gets into a bee's system, it will travel up to the acetylcholine receptors in a bee's brain and block those receptors. This will cause a buildup of acetylcholine in the synapse between neurons in a bee's brain, and it will overstimulate that neuron, and this will eventually lead to paralysis and death for a bee.
Yeah, thank you, Amy. As you can see, we have to be really careful with that. But overall, we learned a lot about honey and why we make it. So I hope you all have taken this lesson to heart. Remember that worksheet is due tonight. Go ahead and finish that up. And then I will see you next lecture. Thank you, guys.